My name is Professor Dorstein. I'm a professor in the School of Pharmacy as well as Department of Pharmacology and Pediatrics at the University of California, San Diego. What I'm really excited about is that one of the things you can do with MASST is you can go actually into the literature, and if there's a MSMS spectra that people have published on, usually in supporting information, you can actually take that spectrum and search it in MASST. And the way you do that is you basically, if you look in the supporting information, you'll see these sort of peaks with an M over Z on the bottom and intensity. And so what you can do is you can take the parent MASST, put it into MASST. It can be pretty rough, but you have to define what search space you're willing to consider. And then you simply go through the spectrum and estimate this mass and that intensity. You do that for the next one, and so you get relative intensities. And so in MASST, what you can then input is the mass and that intensity for all the fragment ions, and then you hit go. And so that allows you to begin searching what other public data sets contains that specific fragmentation spectrum. And I think this is really powerful because there's lots of MSMS spectra where the reference spectrum is never entered into a public domain where you can search it with a computer. And now we have the ability to basically take what's out in the literature and actually try to do searches with it. And so this, the example that we show here specifically are these N-acyl amides. These are ligands that are produced by the microbiome. This was a paper that was published by the Brady group at Rockefeller. And they'd shown that these molecules were able to regulate cholesterol, not cholesterol level, but sugar levels in animal models. And they said that they were clearly associated with the microbiome. However, when we did the search, we found them not only to be associated with the microbiome, but we found that there were many, many different bacteria that can actually produce these kind of molecules. So they turned out to be very common throughout environmental microbes, ocean microbes, soil microbes, as well as human microbiome, rat microbiome, etc., etc. And so to me, this sort of gives a better picture, overall view of the distribution and the presence of these kind of molecules.